plays an very important role in tissue engineering, which aims to fabricate functional tissue for application in regenerative medicines and drugs testing. Tissue regeneration and reconstruction can enable the possibility and repair or replace the damaged tissues and organs. Today, we have Mr. Alok Medikpura, founder of the Nesbic Innovation Lab, with us, and he's a very learned and very expert, distinguished speaker on the theme, how bioprinting is transforming the world. I'm sure that you will have a very active and great session ahead, and you will really get the awareness on the bioprinting. I again welcome you all and thank you for your participation in the webinar. Thank you very much.
Thank you, sir.
Thank you for the introduction, sir.
Over to sir.
Thank you, Chairman, sir. Just to give a formal introduction and to begin with, being a civil engineer, I know even bioprinting has a great role in our line of field also. But you see, we have a very learned professional in bioscience. So, 3D printing has got a great role in civil engineering projects too, and it increases, enhances my interest in the subject very well. So, but bioengineering has its own role, which this subject has come from the head office, and we are very keen to learn about it. And we have got a very learned professional with us, Mr. Alok Medikpura. He is quite associated with the World Economic Forum and has done a lot of research and practice in bioengineering and regeneration of cells and tissues by applying this biomedical by 3D printing. So I would request Mr. Alok to take the charge and enlighten us with his thoughts. Mr. Alok.
Yes, sir. So thank you for the introduction, and it's wonderful to be here interacting with the wider community of engineers who have joined today to know more about what opportunity and potential exists in the field of 3D bioprinting. So I myself am an engineer who has a specialization with automotive and aerospace related streams. And that is when I firsthand got exposure to the wonderful tool of 3D printing and how it could be used to create on-demand customizable solutions. So the whole beauty of 3D printing today is the fact that the entire supply chain of how a product was typically created, mass manufactured, is now changing. And for solutions, whether it is in your aerospace sector, whether it's in your medical sector, in your sort of life sciences, healthcare sector, all of these are not mass-produced solutions. All of these are low volume, high customizable solutions that need to be produced, for which existing mass production techniques of actually manufacturing were not efficient. So with 3D printing evolving, with material science research evolving to an extent where now we are able to create personalized tissues on demand, or even personalized, say, rocket nozzles on demand for the aerospace sector, there's very big disruption happening in all these industries. And today's focus, as mentioned earlier by the fellow panelists, we're going to focus more on bioprinting and the approaches there. So with that, I'd just like to get started with a small presentation that we have prepared for you all. So just give me a minute, please, while I load that presentation. I would like some confirmation on whether you all can see my screen, which has our logo and name at this point, please. Okay, perfect.
Yes, it's quite visible.
So a company we founded about 5 years ago is Nesbic Innovation Labs. And it was founded with a vision to build customizable 3D bioprinters in India. So customizable 3D bioprinters, again, you can think of this as a pure engineering play where you're creating made in India 3D bioprinters. So this is the tool which is necessary to create your bioprinted solutions that we will talk about later. So once you look at creating the 3D bioprinter, then you're looking at increasing the flexibility of the usage of the 3D bioprinter by allowing end users who are, in our case, say researchers who are doing tissue engineering work, researchers who are engaged in the field of regenerative medicine or in cell biology work. So all of these researchers will be working with their own raw material, which we called a biomaterial. So that biomaterial is usually to do with the specific type of tissue that they are trying to maybe regrow in the lab, whether it is a hard tissue or a soft tissue. So it could be a bone-based structure, it could be skin related. So as you can see, the human body is very complex. We have very a sort of a complex structure of cells that make up tissues, and these tissues that make up organs. So while we look at recreating human tissues in the lab, we need to also create the right technology which will enable end users to be able to get the human biology right, the structures, the strength of the tissues right, similar to how a tissue might grow inside your body. That's where bioprinting comes in helpful. And the first step is the tool, which is the bioprinter, which is nothing but an engineering tool, which is custom built and on which we have patents over the technology. So that's what we've been able to do initially, is build custom 3D bioprinting tech. And once the tech was built, we've been able to look at providing this tech to the researchers who have done tremendous work in biomaterials. So collectively, the biomaterials work there, biomaterials work of the researchers plus our strength on engineering is coming together to help create disruptive products in the market. So lab grown skin is something where we looked at, lab grown cornea is something also we are strongly interested in. The area of lab grown meat is something also I'll talk about later, which is something disruptive that can come out of this sector. So for engineers who have joined in today, just treat this as a strong example of how your skill sets of engineering can be applied to various domains. So in the past, we've had core domains, mechanical, electrical, and so forth, but now you're starting to see new trends emerge in the industry, in the job market, where there is requirement for people who know design, who are able to convert that design into solutions such as final parts for bioprinters, or even biotech professionals who know how to use a bioprinter. So you're starting to see a lot of interdisciplinary skill sets that you might need to acquire as a student, as an engineer, to be relevant in the job markets for tomorrow. So with that, we'll just like to get started. So we're a deep tech engineering firm. Like I mentioned, the core focus is the 3D bioprinting technology that we have built in-house, and it's completely made in India, envisioned in India, designed here, and basis on which we are able to create downstream solutions such as the end products, which is bioprinted tissue. So we've named our 3D bioprinter Trivima, which in Sanskrit means 3-dimension. And we're proud to be one of the only engineering firms in India working on this disruptive area of 3D bioprinting. So to also empower the research community out there, we've created a platform called Next Big Learning, which is helping students in their early careers or experienced professionals who are looking to reskill themselves or upskill themselves. With the right approach, we are able to now look at providing through a remote mechanism, a learning module, which will help people get exposure to the field of 3D bioprinting, post which, again, we have a very well engaged community that we have started to build over the years, where we expose the community of people who finish the course to regular webinars, to regular sort of in-person demonstration experiences. So this largely is with the interest to grow the wider research community in India and globally. So that is generally what we would like to focus on. And Next Big Learning, if you just go on our website, there are more details there. So I won't go in depth into that. Second. So some of our sort of versions of the bioprinter that are commercially available today is what you see on the screen. So we have various variants of the bioprinters today, some of which we are commercializing already. So it works from a sort of a academia friendly machine all the way to a professional machine, which in the hands of the right researcher could be used to create disruptive solutions in the area of regenerative medicine and tissue engineering. So this again is a pure engineering play, completely envisioned by Team Next Big Innovation Labs, built, designed in-house, envisioned in-house, and assembled in-house, and commercializing already in the market out there. So for those of you all who are not really aware of how bioprinting works, here is a very short sort of explanation of that. So 3D bioprinting involves, as I mentioned earlier, the engineering as well as the biomaterial side coming together. The biomaterial side coming together is unique to what cell type of interest you're using. So for example, say you want to potentially recreate lab-grown skin in the lab. Then you will look at what type of cells are found in the layer of the skin you're trying to replicate, and then a suitable biomaterial which is also naturally sort of occurring in that specific tissue type is used, and then collectively that mixture is called a bio ink. This mixture sits within a syringe sort of a setup. So here you have living cellular media sitting within a syringe sort of a setup connected to a pneumatic approach through a pressure based system. Then there is extrusion happening, very fine extrusion happening, and you're able to create a three-dimensional structure called a scaffold. So the scaffold that is formed also has live cells embedded inside it. So you're looking at the regular way of 3D printing is just pushing plastic or metal through the nozzle and then getting the end output. Here you're looking at live biological cells going through a nozzle-based system. So that adds to the complexity of printing as compared to other regular 3D printing approaches. So once the scaffold is created, then you move it into the biotech stage where you put it into an incubator, then you give it exposure to the right temperature conditions, the right oxygen conditions, the right set of media changes. And for skin, for example, over a period of about 10 to 12 days, you're looking at the structure of cells growing into the end final tissue. So then this end final tissue is shipped out usually on well plates or on Petri dishes to the final consumer. And who is the final consumer, you might say? So in the early stages of actually adoption, you will find cosmetic companies, fast-moving consumer good companies. All of these companies are looking at moving away from animal testing completely. So whether it's a new soap, cream, shampoo, perfume, anything that is out there in the market, it needs to be tested on animals before it is approved to be put on the shelves. So now that companies have started to realize that animal testing is not just inefficient, but cruel and also expensive to run, these sort of alternatives, such as what bioprinting of skin, for example, can provide, are suitable solutions that they are looking to transition into, which is providing to be really helpful in creating a market for itself. In the long run, a similar methodology will be used to create on-demand skin tissues specific to the patient, which can then be used for transplantation. But we're far away from that, because to get to that stage, we need to get the biomaterials biocompatibility rated by your CDSCO or other regulatory bodies. You will need to get the machine sort of ISO 13485 certified. You need to get surgeons to believe in the fact that this can be put into a patient as a transplant. So that is a journey we are on, and we want to kind of help envision and make India a leader in the space of is the future of bioprinted tissues on demand going for transplantation. We want to make India the hub, India the leader in the space, for which our technology and our work today will help us enable solutions for that market in the future. So why is bioprinting important? One of the key areas, like I said, is as an alternative to animal testing. So why do we need to test on animals when you can test on more ethical, on more sort of viable, cruelty-free approaches that you can create in the lab? So that is one sort of possibility which is available with 3D bioprinting, and which we are also on track to sort of cater to. But more importantly, in the long run is where, like we said, growing bioprinted tissues and organs in the lab, which can be used for transplantation. To get to that stage, there are certain technical as well as regulatory challenges we need to overcome, which we are as a company and as a wider community trying to tackle. So in the meantime, there's also various advantages of bioprinting. For example, whether it is, like we mentioned, that cells extruding out of the machine. So you are able to precisely position the cells within a structure or in a scaffold, which is very helpful to do certain studies, which is cell related and maybe an external drug related, or understanding the cell to cell interactions that happen, whether between a healthy human cell to a diseased human cell. How do you understand that interaction to happen in the lab so as to better understand maybe how, for example, a cancer cell will be affected by a healthy human cell? Those sort of studies you can recreate within the lab. Or to understand the complex tissues, because any organ is made up of multiple tissue types. So how do you understand that complexity of the human organ in the lab, do those studies before you're actually ready for creating transplantable tissues and organs? So this is why bioprinting is important for a community, and this is why it's starting to be spoken about much more deeply, because there is tremendous impact that bioprinting can have on the healthcare sector, on keeping people healthier, on extending their sort of lifespans, and also treating a lot of renal diseases as well. So moving on to some things more engineering related. So is the extruder. So the extruder is important here because, like I said, the core part of the machine is the extruder through which the biomaterials and the cells all flow out and then come onto the end plate of the machine. So there are different extruder technologies that we have developed as a company. One is a pneumatic based extrusion, which we have initially developed. So this is sort of a nozzle based system, which is pneumatically controlled to very good accuracy. And this is controlled through sort of a pressure based technique where you're able to control the extrusion to a very fine extent. So this is one sort of approach we have initially deployed in our bioprinters. Then we moved on to much more complex ways of bioprinting, which is your coaxial sort of techniques and triaxial sort of techniques, which gives you the ability to work with multiple material types at the same time and create very complex structures, such as your tubular structures, for example, in the future, or even vascularization, which is sort of how your blood vessels are carried within the organs inside. So these are very complex structures of engineering that require precise positioning, precise sort of deposition, which solutions like coaxial is able to help us tackle. And also pellet-based extrusion, screw-based. So this is also a unique technology we have developed in-house by Nextbig Innovation Labs, which is helping us work with certain biomaterials which are available in a pellet form, like say your bioceramics or like say materials that are used in your bone. So the raw materials is in a pellet form, and then the end output you need to create specific structures. So this sort of pellet-based extrusion will help you uniformly melt the pellet-based sort of raw materials and then extrude in a uniform manner through the nozzle and then onto the end plate. So this is also a unique sort of engineering innovation from within our lab. Inkjet based bioprinting also is something that we have on our high-end bioprinters. So here you're looking at very finite drop level bioprinting. So this is very critical to certain sort of tissue types, to certain studies where maybe you want to kind of look at creating a healthy human skin tissue and position within the skin tissue certain cancerous cells, for example, to do a study on a melanoma model. So all of these sort of possibilities that exist are only possible when the engineering is able to bring that to life. Inkjet is also one such capability which we have built into our high-end machines. And how is bioprinting transforming the world? I will give you some sort of case studies from not just what we do in the lab, but also what the wider community is doing out there. So you get an understanding of what is really happening in the wider community and how bioprinting is transforming lives around us. So these are all sort of areas where bioprinting is causing a direct impact. whether it is in microfluidics. Later, I will talk about something called an organ on a chip, which is a technology where you create a microfluidic channel within which you print tissue types and then understand better what is the interaction between, like say, the drug molecule and well as the tissue type of interest. So these sort of opportunities are very important for pharmaceutical companies to start bringing down cost of drug development, and then also to help with drug discovery stage being fast tracked. And also like the areas of, for example, cancer studies and simulation in the lab, creating liver models in the lab, which is very critical because every pharmaceutical company needs to test the drug on what effect it will have on the liver. How largely COVID-19 was a big shocker for everyone, but then how the field of bioprinting proved to be helpful in creating lung models. And then the area of skin bioprinting, as well as the area of lab grown cellular meat. These are all sort of opportunities where 3D bioprinting finds its sort of place and can create a massive impact. So one such example is a short video that I would like to kind of demonstrate to you, is the formation of a micro bioprinted heart tissue that researchers at Tel Aviv University created a few years ago. And this short video will explain sort of the idea of how they created it and the impact and the future potential opportunities of what can be created basis on the studies they've done so far.
This is the first time that the whole cellular heart with blood vessel is printed. It's based on a small biopsy that is taken from a patient. The cells and the biomaterial from this tissue are separated. The cells are reprogrammed to become stem cells and then differentiated to become cardiac cells and cells that form blood vessels. And the biomaterial, the collagen and other sugars that in the tissue is processed to become a bioink. And together, when mixed, we have bioinks for the heart and bioinks for the blood vessels. And then we use a 3D printer to print whole hearts with the major blood vessels.
I'm not sure it's going to serve our cell in the next few years, but for our children, I think it's going to be maybe an amazing opportunity.
The whole process may take months or two from the minute that we take the biopsy from the patient till we create the cells and print them. And the next stage is to mature these hearts in the lab for at least a month to help the cells or teach them how to interact with each other and how to provide electrical signal and to have a synchronous contraction or pumping ability of the heart. And in a year or two, we hope to take these hearts and transplant them in small animal models, rats or rabbits.
It will take some time to go from a small sized heart into entire big heart. We still have a long way to go, but we are very optimistic.
So this gives you an example of a technique that we wanted to explain to you called fresh bioprinting or sacrificial, where you see typically what bioprinting methodologies we have shown you earlier is extruding from the nozzle and printing on a flat plate. Here you're printing within a liquid medium by itself. So when you're looking at complex tissues, complex structures like your sort of arteries, your vascular structures, or even something like the heart, which has multiple tissue types, you want to be able to print within a methodology like your fresh and sacrificial bioprinting, because it is easier to create end structures in a format like this rather than just on a final plate, which will not make it happen. And then the support structures also that you will need typically are not necessary when you do within printing within a sacrificial sort of method. So here, this is the advantage. And what you see today is to scale, for example, for your visualization is something like a rabbit-sized sort of heart. So this is a first good step that the entire industry has taken to showcase that we are able to, at a specific scale, create heart-based structures with vascularization within it. So this becomes the base towards us creating more human-like sort of tissues and organs in the future. And moving on, just one second. So another critical thing which usually keeps people in hospitals is sometimes when they face certain challenges with, say, certain muscle damage related to their body. So here at this stage, what really happens is the fastest way to kind of put back muscle into critical areas, whether it's your hand or your legs, which are prone to sort of movements once the patient is completely mobile. You're looking at replanting healthy muscle from a different part of the body into the trauma area. So this challenge is that it increases hospitalization time of the patient, and also increases the costs of actually the surgery, plus the healing of the part from where the healthy muscle is taken also takes time. So to avoid this, the future, what we will look at is actually regrowing muscle tissue within the lab and then transplanting it onto the trauma area. So some work on this regard has been done in South Korea, which we wanted to highlight. And the bioprinting alveoli. So this is also an interesting case study on what has been possible thanks to sacrificial bioprinting techniques, and also say different methods of 3D bioprinting extrusion coming together. So please just watch this quick video. So you can see how flexible the end output tissue is. So this is a miniature lung, as you might call it, and it shows you
research. We're asking, what is the structure of human tissue, and how does that structure underlie its function? Here, what we're trying to do is figure out how to build living tissues made of soft gels and living cells. In the lung, we have the airway and its surrounding bloodstream. What we are seeing here is that the distension of the airway is actually helping to boost the mixing of the blood in the vascular architecture that's flowing around the airway. This is something that may happen inside of the human lung, and it's something that we are exploring more to inform new designs for living tissue.
One of the challenges in our field is making large tissue blocks that have viable and functional cells. And we can make these tissue blocks pretty easily, but the challenge is actually maintaining the viability and functionality of cells inside them. What we're really excited about is making scalable tissues that could one day be implanted into humans.
One of the biggest challenges in making living tissue is figuring out how to get nutrients and oxygen into the tissue and how to get waste products out. The way that our body does this is with vascular networks. But in fact, it's been very difficult to get blood vessel architectures into soft materials in which cells can live. This has been just a big challenge in the field for the past several decades. Now, our lab has been adapting 3D printing technologies for growing soft tissues.
This project was really facilitated and enabled by open source projects like RepRap, Ultimachine, and Grusha. And we're really excited about the ability to also contribute back to the open source community. We believe that science should be open.
When you think about your body, your lungs, your heart, and other tissues are constantly in motion. And so the fact that we're beginning to explore the 3D printing of these dynamic structures, such as valves or dynamic lung-like tissues, is incredibly important for the field.
We are only at the beginning of our exploration of the architectures found in the human body. We still have so much more to learn.
So as engineers, we must have all had exposure to a term called Young's modulus, where you're trying to also understand to a certain extent how, say, materials can bend, materials are elastic in nature. So the whole idea of bioengineering something in the lab also means that you need to achieve the same sort of elasticity of the organ, whether like the researchers have said in the video, achieving sort of the vascular structures, achieving sort of the compactness and the expandability of the lungs. So all of this sort is a material science challenge which we need to bioengineer and recreate in the lab that can be more sort of actual human organ-like. So these sort of challenges, again, as you see how they've overcome it using different methodologies, and where engineering is being quite critical in helping achieve the right solutions. Bioprinted cornea is something which is happening in India, a project that we have been closely watching as well. So Dr. Falguni Pati at IIT Hyderabad, along with his collaborators, LV Prasad Eye Institute, as well as CCMB in Hyderabad, have been working on a very interesting approach to look at actually creating delicate corneal tissue in the lab, which can be used for transplantation and which can potentially be a replacement for certain corneal related diseases, which are quite widely present in the society out there. So the whole challenge here is there are enough statistics to prove that a lot of partial blindness cases in India and certain slight damages to the cornea could potentially find a cure in bioprinting, being able to provide custom patient on demand bioprinted tissue. It's something like a fine contact lens structure that you could potentially be bioprinting in the lab and then transplanting it onto the patient to give the patient a new lease or a new source of life and vision. So these are the sort of research that they have engaged in is quite deep. They've already at an animal trial and research stage. And this is sort of, again, a good example of a homegrown 3D bioprinting project, which is going to make India proud. 3D printed ears. So there are microtia is a sort of a term which is used to depict a birth disorder where the ear of the baby is not really well formed at the time of birth, and it results in something called a condition of an underdeveloped ear. So for two reasons is very important. One, for cosmetic purposes, maybe it's something that needs to be fixed. So you actually have the ear sort of a lobe and structure in place for the patient to not feel left out in the community and to feel inclusive. The second part is that there are sort of results where if the ear is not formed properly, that could also lead to a certain drop in the imparity in hearing. So that could also cause any sort of hearing sort of challenges as well. So to counter that, there has been sort of work done with 3D printed ears that have been created by scientists within the lab, and that has been successfully replanted onto patients. So we've had some of our collaborators in Australia also kind of look at this approach, and even within India, we know a few people who are experimenting on this. So this is again a real-time important problem that bioprinting can help solve. 3D bioprinted bone, again, indigenous solution, and a researcher who's based in IIT Bombay, who's been hard at work in creating biomaterials that have biocompatibility approval. So bioprinted bone, which basically deals with your beta TCP and your HA, hydroxyapatite, is very critical today because certain minor structure damages to the bone, for example, needn't require a lot of hospitalization time and recovery time. If there are solutions like 3D bioprinted bone structures where you can do a CT scan of the patient, understand where there are damages to the part of the bone that needs to be replaced or sort of supported while the bone regrows and regenerates over a period of time, then a small surgery can be done to put that bioprinted bone scaffold onto the trauma area. And then this can help with better recovery for the patient and lead to lesser hospitalization times. Even when it comes to major fractures, where sometimes you will need a medical implant, a medical titanium implant, which is usually the case, that could be avoided in the future when you have bioprinted bone structures put into the trauma areas. And this could also be load-bearing areas like your kneecap, for example. And then over a period of time, as your body regenerates and grows the new bone, then this sort of implanted scaffold degenerates over a period of time. So naturally, as your body is regenerating, this structure is supporting the body in the regenerating process, and then over a period of time, it is biodegradable and just gets flushed out of your system. So that's a beauty in which where we do not need to depend on external structures going into our body and causing long-term implications of uneasiness and certain potential even infections that can kick in sometimes. So all of this can be avoided with 3D bioprinted bone structures once it goes into a transplantation stage. These tissue models, disease tissue models are very critical in pharmaceutical research because pharma companies spend anywhere between 10 to 12 years or about $1.5 billion upwards on developing a new drug that has to come into the market, or even when they're developing solutions which is very specific to maybe a cancerous tissue type or heart tissue type, they would need in lab to understand solutions that can better replicate how an inside or in vivo body inside a human is really is. So how could we help the pharma companies replicate this in vivo environment in the lab? So this is through use of bioprinted tissues along with microfluidic channels coming together in a technology called organ on a chip, which you all can read up about. This sort of an approach will help pharma companies test a drug not against one specific organ type, but multiple organ types. Because once a drug is infused into our body, then there is interaction between multiple organs, and it could affect the multiple organs. It could be helpful for one organ, but it can cause a bad detrimental effect on a second organ. So how do you understand the complexity of a human system within a lab? So that's something where an engineering sort of approach of microfluidics plus the bioprinting approach can come together to create organ on a chip platforms that can help them better study and understand these models. So COVID-19 was a big shocker. We've had one major approach where bioprinting really helped pharmaceutical and the vaccine manufacturers better understand how there is that sort of direct impact of the vaccine on a lung tissue. Here, Wake Forest Institute of Regenerative Medicine, which is a leading regenerative medicine research focused center in the US, built out an artificial lung model, which vaccine companies were able to use to better understand in a short period of time the effect of the vaccine on the lung tissue. Tumor models. So glioblastoma is one of the most prevalent malignant type of primary intracranial tumors out there. And if you can, like an organ on a chip technology, create a solution where you're able to replicate this tumor in the lab, then understand how, the different cell types will interact with this tumor, different drug molecules will interact with this tumor, can potentially help leading to creating solutions for actually treating glioblastoma within patients. This is very critical and sort of affects a large percentage of the population out there. And creating solutions like this within the lab through the use of organ on a chip methodologies will help research community as well as the surgeons better understand how they can be this move towards personalized medicine, precision treatment. So these are all areas where in the future we'll be creating solutions which is healthcare focused to treat a specific individual. So it could be formulated drugs targeted at a specific individual. So we will work with patient-specific glioblastoma samples, patient-specific healthy cells from that patient, and then do studies in the lab. And then once they see positive studies, then go into actual human treatments to treat the challenge. So those were all sort of challenges in the healthcare sector, which bioprinting is playing a critical role in. And one of the other areas is the area of cellular agriculture. So Eat Just is a company, and then there is Aleph Farms, there is there are Avant Meats. So these are all clean meat companies that are popping up out there with the intention to create lab-grown meat. So to create lab-grown meat, it could be either plant-based or it could be cellular-based. So plant-based companies are already out there in the market creating solutions like Beyond Meat, and cellular lab-based companies are still in the late-stage R&D trying to work down the costs and create solutions for the market to have opportunities to consume lab-grown cellular meat, for which bioprinting again plays a key role, because here you're looking at scaling up the production of the scaffolds, scaling up the production of the muscle, the blood vessel, the fat tissues, all of that needs to be grown to replicate the area of interest of the meat arising from the animal. So all of this can be done in a very effective way if bioprinting is used at scale. And also this for us might seem like a luxury here on planet Earth, but it becomes a necessity when you look at space-based explorations, because today on the International Space Station, there is a bioprinting module set up, which is being used to create even say solutions like cellular meat applications. So they've also experimented, as a company called Alep Meats, which is also experimented to create lab-grown meat in space. So this becomes a necessity because you have astronauts who are potentially going to travel about 9 months to get to Mars. That's just one sort of an interplanetary sort of a mission. So how do you keep them well nutritioned? How do you keep them sort of well sort of accustomed to sort of conditions that they had back on Earth, to nutrition that they had back on Earth is very critical. So these are all areas also that are complementary fields where bioprinting can have a very big use case. And how is NBIL in specific helping with these sort of transformations? So we have installations within India, multiple installations, and some of which we'd like to highlight. One is an installation that we have in D.Y. Patil University, where we have one of our bioprinters that is solving a critical problem that they had, which was bioprinting of a very viscous material, which is bone-based. So here, as the material was extruded, you needed to make use of UV-based approach to actually solidify the material. Otherwise, what really would happen is, as the material extrudes, it will not hold the structure that you see is being formed on the plate. So this sort of methodology, our machine was able to tackle for researchers at D.Y. Patil. We've had work done at Narayana Health Institute in Bangalore, which is a center set up by Kiran Mazumdar-Shaw and Dr. Devi Shetty. So here the focus was how could they better do certain studies in the lab, which will help them mimic breast cancer modeling. So here, this was a very early on project where our initial versions of the machine was able to print PCL and gelatin scaffolds on for them in the lab, on which they again seeded the cells and did studies for breast cancer screening. So this was an early stage. Something like your PLCL, this is again a biomaterial which has tremendous potential to be a very good wound healing material. So here the aspect of polylactic caprolactone is that if you're able to create it in the form of like a band-aid strips, for example, and make it stretchable, you're able to use it in critical parts where, for example, there is a mouth cancer surgery that's happened. Post the surgery, instead of putting any other sort of synthetic polymer, if you put polylactic caprolactone, it will help enhance the healing, and it will help attract the sort of the development and the proliferation of healthy cells to enhance the healing process. So here again, this was a critical experiment that we did for one of our clients, and this showcased how our bioprinter could be used to create an end product here, which is the actual elastic PLCL material. The raw material again was here in pellet form, so the conversion of that into the end output was a challenge which we were able to tackle. Some work we have done in Big Pharma was at Merck Group in Germany. So here Merck Group is one of the largest and the oldest pharmaceutical and life sciences companies based out of Germany, 350-year-old plus. So there we went through a program where they globally identified all the options for bioprinters out there and chose what we at NBIL had manufactured. And then through a process of about a year, we did critical experiments with their drug development team, with their sort of cancer research team, and other teams within Merck to demonstrate to them how we have created viable engineering tech which can do critical things like do droplet level printing within 384 well plates, which is both accuracy and efficiency and a time-based challenge which we're able to tackle for them, and also go into a more complex structure of creating a drop on a drop model or two drops next adjoining each other model. So all of this was to showcase how engineering can be fine-tuned and improved to bring more accuracy into tech and bioprinting processes. So typically all of this was done, would have been done by scientists in the lab through a certain method called pipetting, which is very inefficient and inaccurate and will not give you good reliability, and it will not give you good repeatability. But a solution like a bioprinter is able to be used there to create that repeatability, which is very important for any sort of experiments that they do in a pharmaceutical setup. And with that, I just like to kind of stop at this point. We've covered quite a bit, and I know you all are mostly from an engineering background. So, but just so eager to know if you'll and hear from you all about any questions you all might have.
Yeah, thank you, Alok. It was a nice presentation. You have covered almost all parts of human body and the application of your machine. What I have learned in my own field of work of 3D printing. So there's a 3D machine from which a fluid or a construction material comes through a nozzle and forms a structure with as per the design on a computer controlled designing system, CAD or something like that. So the same thing I saw in your presentation in the machine which you have set up at DY Patil University. So I would request you that the same kind of one, you make one video on the other parts of the thing which you have produced, how the machine actually helps in the applications you have mentioned. So, and secondly, would you please tell us what's the difference between robotics and 3D printing or bio 3D printing?
Sure. Thank you for the feedback, sir. So we're definitely working on creating more content which will help the first-time visualizer, first-time user of bioprinting also understand the various aspects involved. So we're in the process of doing that. So your second question on robotics and bioprinting. So typically you can look at the 3D bioprinter as a sort of a level of robotics as a play in that, because anything to do with robotics, you're looking at system-wide control, and you're looking at programming the robot. You're looking at the equipment in terms of the machine, in terms of the use of, they say, the robotic arms. Here you're looking at our XYZ axis, which is moving. So it is not a 6-axis sort of a typical human robot, but here we're deploying the same methodologies of robotics. In fact, we're going to the extent of building our own board out. We have our own software that we have developed to control the machine. So all of this is the same backend that goes into someone learning robotics. So that approach, along with our approach of actually creating our own software, which is very relevant to engineers in the computer science field, our also approach of actually using artificial intelligence-based methodologies to make bioprinting much more smarter. We have an ongoing project with Microsoft there, is something unique. Mechanically, we're designing all the parts ourselves using Autodesk software and machining it, and then manufacturing and then installing it. So that is again an approach of how a designer can find a use case with bioprinting. Mechanical engineers find a use case, and then a lot of the studies, whether it's flow control or sort of better understanding the material science angle behind things, also can be done remotely with certain tools which our team is using. So this gives you an idea of how there's an interdisciplinary mindset and a team behind actually creating solutions in bioprinting.
Okay, thank you. So they have so many questions coming up from the participants, but we are running short of time also. I would request Mr. I.S. Oberoi sir to see if the time permits, then I can take up the questions.
Yes, sir, you can take the questions. We have the time. We have around 15 minutes. You can take the questions, please.
Okay, thank you. Sir, one of my participants asked how much this technology has developed and adapted by the other parts of the world or the whole world at present?
Sure. So if I have to answer 3D printing, I can tell you 3D printing is widely used in today aerospace. Like there are your Elon Musk company SpaceX is building their own custom rocket nozzles. The same thing is being done by Rocket Labs, and other company. There's another US related company that's looking at building out the entire rocket using metal 3D printing. So that application is there. Automotive, they're using it widely to do a lot of studies and R&D in computational fluid dynamics side, CFD, wind tunnel studies, which is something I also did while I was at university. In the medical sector, custom 3D bioprinted implants are being printed today at ISO 13485 facilities and are being used by patients. So bioprinting and biotech areas are being impacted largely in the research area right now. In the next 5 years, you'll see a lot of commercialization of this coming into mainstream. And India is also doing relatively good work in all of these areas. And I would say for bioprinting, it's our goal to make India the hub and the leader. And we want to create disruptive solutions in India that will not just be renowned for the work here, but also meet global standards and get global accreditation.
Okay, thank you. One of my participants, Mr. Ritik Satpute, has asked, is 3D printing is something similar like that cloning technique or technology?
Cloning is a different tech. So when you look at cloning, you go into like the DNA stage. You should be, if you're interested in something cloning related, you should be looking at like say cloning, like say Dolly the sheep was one of the first animals to be cloned. So there you're looking at replicating the DNA and then moving into recreating the same organism separately. But when you look at bioprinting, what we look at is re-engineering the tissue. So we know how a human tissue is. We know what cells are in the, for example, a top layer of the skin. We know that the top layer of the skin forms a specific purpose in our body, which is to ward off external agents. It has a certain elasticity around it. It has a certain functionality where if a product sits on it, it absorbs into it. So how do you recreate this in the lab? That's basically called tissue engineering, because you're engineering the same tissue in the lab. And why do you want to re-engineer it? Is maybe because you need to better understand how a new soap that is created will have an effect on the skin. So you want to do those studies in the lab instead of doing a study on an animal. Or in the long run, you might want to create a bioprinted tissue which is specific to an individual, because maybe there's a case of a burn. So instead of actually taking skin from a different part of a body and then putting it into the trauma area, you might want to bioprint skin on demand and then transplant it onto the patient. So for those use cases, we will do bioprinting, and it's the area of tissue engineering and regenerative medicine as more is widely called.
Okay, thank you. So another question coming from Debashish Naik. He says, for the cells, you might be selecting the materials. Do you characterize them by yourself or seek the materials data from other people?
So the basis of how we collect the cells is one, it can be coming from a patient specifically. So for example, like say, there are cell banks. So cell banks out there, you can go purchase cells from them, and then you know that cell has come from which part of the body, how old the patient was when the cells were collected. So all of that data is very helpful, because cells from an older patient mean that the cells have this functionality of not doubling and becoming more number of cells, because that proliferation rate is lower. Cells from a younger patient means the proliferation is better. So certain characteristics like your cellular based studies we in turn have to do, and then also the biomaterial that needs to be used also we need to be utilizing, looking at the source, because it could be naturally derived, it could be sort of synthetic biomaterials. So those sort of options are available and need to be looked at for whatever use case we are looking at. For example, when we are looking at recreating skin in the lab, it can just be human skin. It doesn't matter if it is specific to me or you as a patient. But when I'm looking at maybe creating transplantable skin onto someone, it needs to be very patient specific, because you don't want the body of that patient rejecting that sort of a transplant in the future. So then you go more detailed into the patient biology.
Okay, thank you. Another question is, do we produce a new cells from single one? I couldn't understand this question actually.
Yeah, so typically what I would like to explain to is the way cells are. So say when whether it is the field of lab grown cellular meat, where they're saying I can grow lab grown meat. What does that mean? That means you take a small biopsy, meaning you take a small syringe, you put that into an animal, and then you collect like certain muscle sample of the patient, of the animal, for example. Then that will be in a small quantity. Cells have this functionality where they grow. So that is when you give it the right medium, you will get the cells to double, triple, and keep like sort of dividing and growing. So then that is how you make from a small collected sample a larger sort of volume of cells in sort of tens of millions, and then you will put them, you'll store them in a cell bank. In the cell bank, usually it is stored in a liquid nitrogen tank and it is frozen. At the time of requirement of using the cell, then you will bring it out of the liquid nitrogen tank, revive it, and then use a specific portion of the cells for the printing. So that is the process. I hope that was the question that you have, whether you take a single cell. It's not a single cell, it's maybe a few. I'm not able to give a number because cells are like sort of micron accuracy. So
Yeah.
you can consider maybe if you take like a few million cells, you're able to go into few 100 million cells.
Correct. So what do you see? What are the key challenges and limitations of bioprinting technology is be seen today? And what are the software used for the 3D bioprinting? Is it similar like Autodesk, Fustian 360?
So the software, the Autodesk part that we use mostly today is for the mechanical work that goes into the bioprinter. So everything you see in the bioprinter that I showed you earlier is all work of us. It's all something that we have developed in-house as a company. So everything from the design, outer design, to the structure of the machine, to the extruder part, to the bed, everything is designed by us. So here we're using Autodesk. So when you go into design, into like the development of the scaffold, which is the end output when the printing happens, the structure, that is created by us using other sort of softwares. And the methodology of actually getting the accuracy right is sort of dependent on the engineering code that goes into running the machine. So it's something called a G code that controls the machine to a specific accuracy and efficiency, which we are able to do. So that was the answer. I forget the other part of the question.
It was only asked about the software being used. And there's a one very curiosity of one of my participants is how this, what is the scope of 3D bioprinting as a career?
Sure. So like I said, 3D bioprinting has tremendous opportunity in terms of impact, because, for example, you could be a mechanical engineer and you find a role in actually developing the bioprinter. You could be a material science researcher or engineer. So you find a role in actually becoming a bioprinting, biomaterial specialist. You could be someone who has experience in CFD or other sort of flow related specific skill sets. So then you will have a use case to better understand the flow rates and replicate within an in silico method, and understand how well bioprinting can be tweaked depending on the nozzle sizes. So different sort of approaches, different sort of fields of bioprinting have use cases. And one such sort of opportunity for you all to better understand this is through our next big learning platform, which we have created. I'm not able to find the chat option, but if you'll just go Google Next Big Innovation Labs, and then the website is NEXTBIG, what you see here on the screen. One second. So nextbiginnovationlabs.com, and then there is an option called Next Big Learning. So where you are able to better understand what sort of careers can come out of 3D printing. We brought in industry experts to come talk about that. We brought in researchers, we brought an academic institution also to be part of the course. So that if you're interested, can be a good stepping stone towards then deciding whether you'll want to create any solutions. I mean, you'll want to kind of participate in becoming a key stakeholder in the field of bioprinting.
One of my participants, Akbar Shah, he wants to know whether bioscaffolds developed from bioprinting can be used in repairing fractures.
Yes, definitely. So bioprinted bone-based structures, when used in the future for treatment of bones, could help treat fractures as well as certain sort of hairline fractures, all the way to even maybe broken bones, could be replaced with bioprinted bone-based structures. So today the use case is that you have use of a lot of medical implants. So instead of using a metal medical implant, which in the long run will become very uncomfortable for the patient, you could be depending on bioprinted patient-specific implants. So how that happens is, say you have a fracture. What you do first is go get a CT scan. In the CT scan, the doctor is able to visualize how bad the fracture is, whether it's a broken bone or it's only a slight sort of hairline fracture. From that, then you're able to derive certain data and then print out in the lab bio-compatible 3D bone-related structures, and then put that into the patient. And over a period of time, as the bone regrows, that material that you put into the patient will sort of biodegrade, and then also help in assisting the regeneration of the bone as well. So that is the future we are going towards, which will be very targeted to the patient and the individual.
Okay. Thank you very much. There are so many number of questions which are almost similar in nature, and you have already addressed to. I would like to close down here, and will request Mr. Oberoi sir to take over and conduct the program further. Thank you.
Thank you, Bajaj Saab. It was a wonderful session, Alok, and I would just compliment your company and your vision towards making India self-reliant in bioprinted products. And I believe that, yes, the future is there. Another couple of years, if we have, I think we can, it can be a technology disruption, and it's a wonderful concept which you guys are promoting. And I would just now go in for the vote of thanks for this. Good evening, everyone. Our respected chairman, sir, Engineer K. Kuldeep Raj Gupta ji, our expert speaker, my fellow FLCC committee members, and all the audience present. I, on behalf of Institution of Engineers India, Faridabad Local Center, take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to this webinar on the topic how bioprinting is transforming the world. I would like to express my sincere gratitude to Mr. Sudeep Chakravarti, Mr. Kingshuk Sen from IEI headquarters and their team for their support. My sincere thanks to our expert speaker, Mr. Alok Medikarpura Anil, founder of Next Big Innovation Labs, for making excellent presentation and making this webinar interesting and meaningful. My special thanks to the event moderator, Engineer Sushil Bajaj ji, who has been always, he is a powerhouse of knowledge. And once we started this conversation, he said, yes, I am not conversant with bioprinting, but I think, Bajaj Saab, you really handled the total show wonderful. So it's a wonderful, my special thanks to you for your unflinching support and making this event a resounding success. Last but not the least, I would like to thank all our II honorable members and all the participants who have blessed with their presence and took valuable time of their busy schedules. So your presence and participation has made this event up to the mark. For the participants, I would like to mention one more thing, that in case you opt for an e-certificate of the participation, you can register on our link which was given in the flyer and make a nominal payment of 300 rupees plus GST using the payment gateway. This link against the link, and this is valid till 1st October 2023. So you got one full day. So you can, if you need, you will you can have the e-certificate on the payment of a nominal amount of 300 rupees. With this, I would go to the end of today's webinar. I would request our event moderator, Engineer Sushil Bajaj, to kindly take the procedure and close the webinar, please.
Thank you, sir. Thank you very much. And with this, we close the program. And thanks to Chairman, sir. Thanks to all the dignitaries from the headquarters. Thanks to our Secretary, Mr. S.O. Brai, sir, and all the participants who joined with us. And attentively, there were 52 participants from the beginning and remained there, nobody leaving in between. The program was so interesting and engrossing. Thank you very much.
Thank you, everyone.
Thank you. Thank you very much, sir. I would request headquarters to kindly close the webinar, please.
Thank you, sir.